I'm just going to take a minute to introduce our very own Mukesh Guatam, who's a research assistant professor of neurology. Mukesh has been had a long-standing interest in investigating mechanisms of vulnerability and that render motor neurons particularly vulnerable in the context of ALS. And I'm delighted that he's here today, and he's going to tell us about this exciting story on the utilization of these nanoparticles that may be of therapeutic relevance in ALS. So thank you, Mukesh, for being here.
Thank you so much, Dr. Evangelos. Good morning and welcome to the symposium. I'll be talking about the strategies that we take to improve the health of vulnerable upper motor neuron by improving the mitochondria of these neurons. Upper motor neurons are very important. They complete the motor circuitry by making connection with the spinal motor neuron, spinal cord. And in ALS, along with the spinal motor neurons, upper motor neurons also degenerate. So why TDP-43 is important, and Dr. Kiskinis and Dr. Bosco has beautifully explained in detail in the previous presentations that TDP-43 is critical in ALS. Also in frontotemporal dementia and in the upper motor neuron diseases such as hereditary spastic paraplegia and primary lateral sclerosis. TDP-43 regulates many aspects of the RNA metabolism, as well as cellular metabolism in ALS, and more relevant and important for us is the mitochondria dysfunctions. TDP-43, when it translocates to cytoplasm, it also goes and binds to the mitochondria, and it binds several molecules in mitochondria, which are important for the oxidative phosphorylation, as well as the autophagy and the mitochondrial DNA functions. In our previous studies, we've shown that TDP-43 using postmortem human brain from TDP-43 patient with TDP-43 pathology, that the upper motor neuron, or the bed cells that we call, they have mitochondrial dysfunctions, and particularly the inner mitochondrial membranes disintegrate in these upper motor neurons, whereas the outer mitochondrial membrane largely remains intact. And this pathology is not seen in the other non-bed cells in the motor cortex in the patient. Interestingly, we see pretty much the same pathology in the mouse model of TDP-43, whereas in this upper motor neuron, the outer mitochondrial membrane remains intact, but the inner mitochondrial membrane disintegrates. And so what is so important or so interesting about the inner mitochondrial membrane? So inner mitochondrial membrane is the place where all the proteins that are important for the energy generation and the metabolism are located. And these membranes are made of phospholipids. And one of the important phospholipid is cardiolipin. Cardiolipin is a phospholipid with a phospho head and 4 lipid tails. And because of it, it gives us a special ability to cardiolipin to make the curvature of the inner mitochondrial membrane that makes the crystal structure in mitochondria. And as you can see on the left-hand side panel, that cardiolipin, it regulates many aspects of the mitochondrial function, such as the fission of mitochondria, the energy generation, and also the leakage of the mitochondrial DNA and other molecules from mitochondria. And so what we could do to help this situation, to improve the mitochondria structure and function, and that may in turn improve the upper motor neuron health. And we use 2 strategies. We use 2 small molecules. One is the small molecule called SBT272, and the other approach is the nanoparticle that has cardiolipin in them. For the first approach, we collaborated with a company called Stealth Biotherapeutics. And they have this small molecule called SBT272. This molecule binds to the cardiolipin and stabilizes the cardiolipin structure. And to investigate further, what does it do? We use this mouse model called PTDP43-UCHL1 EGFP mouse model. In this mouse model, the upper motor neurons are green by EGFP expression. And for our studies, we use this mouse model at 2 time points. It's 3 days old and P120 days old. The 3 days old mouse we use to culture these neurons in vitro and then perform our studies in vitro with these molecules. As you can see, the wild type neurons do not have TDP43 in them, but in the mouse model, they express the human mutated form of TDP43. And so we performed this technique, electron microscopy technique called correlative light electron microscopy, in which we can select a neuron that is GFP expressing, that's the neuron of our interest. And then we do electron microscopy to see what is going on inside that particular neuron. And in panel B primes, you can see in the wild type neurons, the mitochondria look good in number, and they also have in a mitochondrial membrane intact, and they are not clumped together. But in the diseased neurons, that is C to C primes, you see again, similar to what we saw in the mouse model and the human tissue upper motor neurons, that the outer membrane, mitochondrial membrane remains intact, but the inner mitochondrial membrane disintegrates. Additionally, these mitochondria come together and they form clusters or clumps together. But when we treated these neurons in vitro with SBT272, we can see the regeneration or the protection from degeneration to this inner mitochondrial membrane, and also the mitochondria did not clump as they did in the diseased model. And these are the high mag images showing that we have better inner mitochondrial membrane intact here. And so we perform the functional assays for the mitochondria function, which is treat them with TMRE and do flow cytometry analysis. And the TMRE accumulation shows the polarization of mitochondrial membranes. And you can see that after the treatment with SBT272, we have more mitochondrial polarization that shows the functional ability of this mitochondria. There's another outcome measure that we take is to see how the exon projection of these neurons are in vitro. And when we treated these neurons with SBT272 plus the other drugs that are approved for treatment of ALS, Adenovone and Amylyx, and we see that the SBT272 improves the axon length of these neurons, as well as the arborization of their neurites. And so, encouraged by these results, that in vitro results, we decided to go in vivo and see does SBT272 improves the mitochondrial health and the upper motor neuron health in vivo in this mouse model. And for this, we started to treat TDP mice with SBT272. We used 2 doses, 1 mg per kg and 5 mg per kg for 60 days. So we injected the mice with SBT for 60 days, starting P60 up to P120. And then we analyzed the CSMN or upper motor neuron numbers and the gliosis in the motor cortex. In the wild type, we don't see gliosis, and there is a good number of upper motor neurons, whereas in the TDP mouse, the motor neuron numbers decreases and there's massive astrogliosis and microgliosis. And interestingly, with the treatment of with the SBT272 at both the doses, 5 mg per kg and 1 mg per kg, we see a significant reduction of the astrogliosis and microgliosis, and also the number of upper motor neurons are increased. So SBT272 seems to be an effective small molecule that improves the health of upper motor neuron and decrease gliosis. The other strategy that we take is to use these nanoparticles. We collaborate with Dr. Shad Thaxton here at Northwestern. Dr. Thaxton has generated and characterized these nanoparticles. We call them high-density lipoprotein nanoparticles. There is this 5 nanometer gold core, and then there are these apolipoproteins on the surface. And the surface of this nanoparticle is loaded with 18,2-PG phosphatidylglycerol and cardiolipin. As I said earlier, these 18,2-PG phospholipids and the cardiolipin are very important for the mitochondrial membrane structure, integrity, and function. And so we think that if we supply these lipids to these vulnerable neurons, it might improve the health of mitochondria and the neuron itself. One important feature of these nanoparticles is that they only bind to the cells that express SRB1 receptor, and that gives it the ability to target these nanoparticles to the specific tissues. And as I showed earlier, that inner mitochondrial membrane is lost in the upper motor neurons, and giving them more phospholipids and cardiolipin might regenerate and help them. So we tested if the SRB1, that is the target for these nanoparticles, is expressed by the mouse upper motor neuron and the human upper motor neurons. And in the panel A, top panel, you can see the SRB1 is expressed by the GFP expressing upper motor neuron, as well as the MAP2 expressing best cells in the human motor neuron. The cortex. And these nanoparticles, this is the assay to show that these nanoparticles do indeed transfer their cargo to their recipient cells. Here, the nanoparticles, these cells were treated with this NBD cholesterol, and in the merge, you can see that cholesterol was transferred to the cells of the interest. So it seems that these nanoparticles are able to deliver their phospholipid cargo to the target cells. And in a pilot in vitro experiment, we show that treatment with this cardiolipin nanoparticles, it does improves the axon length of the upper motor neuron that are diseased with TDP43 pathology as compared to the one which did not receive it. And so we wanted to see if it does, what does it do in vivo in the mouse, in TDP43 mouse. And we took similar strategy where we used the TDP43 UAGFP mice for these experiments. And here, this time, we started our treatment at P60 because these mice start showing symptoms at P60. And this time, we treated the mice with the nanoparticles 2 times a week, 500 nanomolar in 100 microliter. And then we had a saline treatment mice as control, and then tried to see their similar what we still did for the SVT2 to see the CSN number and the gliosis. And interestingly here, the TTP mouse shows a lot of astrocytes and microglia in the motor cortex and reduced number of upper motor neuron. But with the treatment with nanoparticles, it improves the, there is more CSMN in the motor cortex and significantly lower GFAP and the microglia upon the treatment. So in summary, we can say that the upper motor neurons have profound mitochondrial defects in ALS, and that the pathology that we see in mouse upper motor neuron and human motor neuron is similar. And improving the inner mitochondrial membrane, it could be an important strategy. And improving mitochondrial health can indeed improve or provide neuroprotection in ALS. And modulation of lipid homeostasis could be another strategy to provide the neuroprotection in ALS and FTD. And I thank Dr. Hande Osdindler for all her help, support, and guidance, and my wonderful lab colleagues and funding from Spastic Paraplegia Foundation and Revelio Inc. and Illumina Pilot Grant Award. Thank you very much.
Thank you so much, Mukesh. Maybe we have time for a couple questions. And I must say thank you so much, Mukesh, for being with us, given the circumstances. and we appreciate you being here. Thank you so much. There is one question, maybe I can read from the chat.
Yes, please.
Hi, is there any role of a peroxinase enzyme in neuroprotection? Rahul Saxena, Sharda University. Peroxinase. Maybe this is a question for you, and it could be also for Daryl Bosco.
Can I ask a question?
Yes, maybe you know the answer. Peroxinase enzyme. All right, maybe we'll write after the meeting. All right.
That was really interesting. Thanks for the great presentation. So maybe I missed something at the beginning, but my question is, okay, I'm going to phrase it differently. In what subtypes of ALS would you expect?
Sorry.
In what types or subtypes of ALS, as we heard about, for example, the genetic subtypes, might you expect to see this type of pathology? Is that something that's known from any clinical data to see mitochondrial ultrastructure just?
So the patient tissue that we use for the studies was for the ALS patient with TDP-43 pathology, also the FTD patient with TDP-43 pathology, and we see pretty much the same thing.
Okay.
But so in the other study, we've used that was not in the patient tissue, but in the mouse tissue. And we use, in the SOD one, we see totally different than what we see in the TDP.
Okay, so that's really interesting. So they're both, the SOD1 model and the TDP model that you used are both kind of overexpressing a lot of mutant protein. So I thought, oh, maybe it's a general thing that happens downstream of problems with protein accumulation. What do you think?
Yeah, I would say so. So like in the SOD one, but that I cannot exactly say that it's exactly same what we see, because in that study we looked at P15 neurons in the P15 mouse, which is very early. So even at P15 we saw similar to what we see in the adult mouse in TDP-43, but in the SOD1 they were totally different, like very large mitochondria, and they were maybe trying to compensate that time. But I think to answer your question best, I should have the data right at the same age where we can see,
In the SOD1, this was also motor cortex, not spinal cord, right? You were looking at the BET cells in the SOD1 mice?
Yes.
Okay,
yes.
Thank you.
Yes, thank you.
Hi. Do you know whether SBT or these particles that you're working with actually pass the blood-brain barrier? I mean, maybe they're working peripherally. Maybe they're improving lipid homeostasis.
Yes, very, very good and pertinent question. SBT2, it does. So we have data about it. The nanoparticles, we are working on it right now to see if it does cross it. So we are trying to do the biodistribution study. But the other aspect, or there is another hypothesis that we think is the SRB1 receptor is expressed by the endothelial cells in the brain as well. We've done some studies. And the thinking is that these nanoparticles, even if they don't cross, the gold code thing does not cross the blood-brain barrier, but when it binds to those endothelial cells, it transfers the cargo to those cells, and that's how it gets into the brain. That's our thinking. We don't have definitive data as of now, but we're looking into it. Thank you.
Hello. Does increasing HDL in a patient's diet improve mitochondrial protection, or if it is just reliant on nanoparticles?
Good question, very good question indeed. But if we take it but in the diet, then it goes into the system, and this processing is very different than what it would be to the targeted delivery. I don't know exact answer to this question. Yeah. Thank you very much.